it requires chaperones. This is to show you at the nuclear pore how dynamic this process is. Some proteins go in, like ribosomal proteins, that are being translated outside of the nucleus at the cytosol. These proteins travel through the nuclear pore, get inside, they associate or bind to RNA or pre-RNA, ribosomal RNA that is being made from ribosomal DNA, which is localized at the nucleus. This complex also suffers from cleavage and modifications. It turns into a 18S and the 28S and the 5.8S. This is a sedimentation kind of designation. 5S that comes from outside. These two subunits, the ribosomal subunits, is made out of all this, the RNA, the ribosomal proteins. They translocate out. They get out. They form the 40S subunit, the 60S subunit, and then they associate outside. So there's a lot of complexity in the making of the ribosomes. And it's made, conformed by RNA, protein, etc. All this happens through the nuclear pore. So, recapitulating the nucleus. There is the DNA in the form of the chromatin. The euchromatin, 90% of the genes are, for the most part, active. Okay? It all depends on the cell type and the stage of at which the cell is. But when you see euchromatin is lax and most of the genes within the chromosomes are active. Then you have some percentage of the chromatin that is very compact with the histones and other proteins, and it forms the heterochromatin. And it's associated to the inner part of the nuclear envelope. Then you have the nucleolus, where the RNA is processed, like mRNA, messenger RNA splicing, ribosomal RNA maturation, as you've seen, and assembly with ribosomal proteins, etc. So this happens at the nucleolus. Then you have the nuclear envelope, which are made out of two consecutive membranes with distinct composition and function. For example, the ribosomes attach to the outer membrane, right? And there's a lumen in the nuclear envelope inside, which is connected to the lumen of the ER. Plasma membrane, just to give you an example. Plasma membrane is made out of only one membrane, while the nuclear envelope is made out of two consecutive membranes. Okay? Plasma membrane doesn't have a lumen inside, just one membrane. This is different. The inner membrane is associated with the lamina, which binds to the heterochromatin, right? The densely packed chromatin. The lamina segregates with phosphorylation during mitosis, and that leads to the segregation of the chromosomes and the disintegration of the nuclear envelope. The outer membrane is associated with the ribosomes. And just recapitulating what I explained. The nuclear pores are involved in bidirectional gating. This is gating transport. It is made out of a very large protein complex formed by 50 nuclear porins. There are 3,000 to 4,000 pores in the nuclear envelope. The nuclear pore is very active. For example, 100 histones pass through the nuclear pore every minute. Small proteins pass through the fusion. Large proteins require active transport via chaperones, GTP and ATP hydrolysis. And the nuclear proteins, the large ones, they are nuclear localization signals. Okay? And they are easily identified when we sequence one of the proteins. If it has that recognition site, then we know that this protein translocates into the nucleus. Right, mitochondria. This is a schematic of a mitochondria. We again see two membranes. One is the outer membrane, another one is the inner membrane. There is an intermembrane space. It's very complex and it is involved in lots of activities. Most important one is to provide the cell with energy, respirate. Okay? It's responsible for the respiration of the cell. It has invaginations. The inner membrane has invaginations called the crista with the intracrystal space. And then we have the matrix here, the mitochondrial matrix, or intercrystal space. Here we have the DNA. The mitochondria has DNA, its own DNA. And we'll see that over the course of the lectures. Has enzymes for lipid and protein synthesis, and is responsible for the Krebs cycle. It has all the enzymes. This DNA bears a few genes, but there are many more proteins within the mitochondrial structure. So how can this happen? So this can happen only because there's a lot of protein import from outside, from proteins that are being made in the cytosol. They translocate into the mitochondria, and they form all the complexes required for the synthesis of ATP. There are mitochondrial granules, there are cation binding sites, and this is a magnification of the inner membrane. These complexes are involved in the respiration, in the electron transfer process. And these are made out of enzymes for oxidative phosphorylation and for ATP production. Okay. These two membranes, the outer membrane and the inner membrane, are very different. And the explanation, the evolutionary explanation for that is that only the inner membrane was part of a bacterium that was engulfed. And that's why it looks so different from the outer membrane that was part of a cell, part of the eukaryotic precursor cell. This is another schematic. And again, whenever you see lots of mitochondria, that means that there is a lot of energy involved in the form of ATP utilization. For example, this is mitochondria in a kidney tubule cell that accumulates at the base of the wall, where active ion exchange to specific pumps require ATP. So whenever you see mitochondria, for example, in the skeletal muscle, or here at the kidney tubule, that means that there's a lot of activity, but a lot of energy involved. So the mitochondrion contains its own DNA that produces enzymes involved in ATP formation. Most of the proteins, or another set of proteins, is imported. Protein import occurs post-translationally. So first the protein is translated, and then it is imported into the mitochondria. It's different from the co-translational import, which happens when the protein is being made and translocates into the ER. ATP, or energy, and chaperones are required for protein import into the mitochondria. The mitochondria uses a lot of oxygen to produce the ATP. The peroxisome also uses oxygen, but the difference, and we'll look into the peroxisome briefly later. The difference is that the oxygen consumption here at the mitochondria is coupled to ATP production. So the mitochondria respirates to produce energy. Okay. Then there is one aspect here, the cytochrome C, which is embedded within all these electron transport complexes here. The cytochrome C plays, it's a very small protein and plays many different functions, important functions. So all the proteins of the electron transport chain are organized in 4 multi-protein complexes that cross the inner mitochondrial membrane to extrude protons from one side to the other. So all this extrude protons from one side to the other. The only exception is cytochrome C, very small protein, 12.5 kilodalton, or 100 amino acids, very, very small compared to the others, that is loosely attached on the outer surface of the inner membrane. This is the inner membrane of the mitochondria, and this is the cytochrome C. Structure allows cytochrome C to move freely in order to transfer electrons from complex 3 to complex 4. This is complex 3, this is complex 4. In case of mitochondrial damage, cytochrome C is easily released and can migrate to the cytosol, which is the main trigger of the mitochondrial pathway of apoptosis. It's like a trigger and a checkpoint. Whenever there is mitochondrial damage because of extreme conditions, consumption of energy, the mitochondria is prone to die, then there are ruptures, there is damage of the outer membrane, so the cytochrome C that is loosely attached can go out of mitochondria. That triggers the mitochondrial pathway of apoptosis. So it's a very clever way of trigger cell death. Whenever there is damage of the mitochondria, there will be cell death for the most part. And cytochrome C is responsible for that. So now the secretory pathway. see a cell, for example, that has these attributes here, whenever you see all these vesicles here, that means that this cell secretes products outside of the cell. Okay? So the structure will tell you a lot about the function of the cell. So this is the Golgi apparatus, which is connected to the ER, or the plasmic reticulum, through vesicles that go back and forth from the ER to the Golgi. Golgi is also very complex, and it looks like a stack of pancakes. And it's been formed by the ER-Golgi intermediate compartment, the cis-Golgi network, the trans-Golgi network, and the Golgi stack or Golgi proper, the medial and trans. And from the trans-Golgi, lots of secretion vesicles are budding off. And these contain all the information, all the required information to go to one way or another. This is the ER. The ER is intimately connected to the nuclear envelope. This is the nuclear envelope with the lumen here, with the outer membrane here with the ribosomes, and this invagination that forms the ER, the plasmic reticulum, with the ribosomes. When you see, for example, this structure, and you see the ribosomes on both sides, this could not be the nuclear envelope, but it could be the ER. Ribosomes, we have a common pool of ribosomal subunits in a cytosol that could be utilized to make a free cytoplasmic RNA that will give rise to a cytoplasmic protein, or it could make also a protein that is prone to translocate into the ER. Intriguingly, the ribosomes are not different from one or another. There is a common pool. The information that makes these two sets different is located in the nascent protein, which has an ER signal peptide that will allow this newly synthesized protein to attach the whole complex to the ER. So in sum, the ribosomes are the same ones, but what contains a distinctive information is in the newly synthesized protein. Here's an example. So we have the two subunits, the small and the large ribosomal subunits. The mRNA, the tRNA, the transfer RNA, the new protein that is made out of the addition of the minimal components or the amino acids, a recognition signal here that creates like a channel within the endoplasmic reticulum and leads to the attachment of the ribosome to the ER. So the information is contained in the new protein, not in the ribosome. And this is the lumen of the ER, or the cavity of the ER. This is an electromicrograph of the ribosomes here, which are free ribosomes that are hanging in the cytosol, and these other ones that are associated with the ER, as you can see here. These ribosomes are exactly the same as these ones. The difference is in the protein that is being synthesized. So, the endoplasmic reticulum, we have soluble and ER bound ribosomes. These are identical. An ER signal sequence in the protein is recognized by a signal recognition particle, which allows for the co-translational translocation into the ER. Co-translational because at the same time that the protein is being made, it translocates into the lumen or into the ER. Hydrophobic proteins are anchored to the ER membrane. Proteins to be secreted are translocated into the ER lumen. It all depends on the nature of the new protein. If the protein is hydrophobic, it will be attached or anchored to the ER membrane. But if the protein has an ER signal but is not entirely hydrophobic,